Thank you. I am delighted to be here again this year and to speak on this talk. I'm going to be speaking on the current and future role of precision cancer medicine and targeted therapies in female pelvic malignancies. Now, that's the title. I am going to be focusing essentially exclusively on ovarian cancer because that's where the data are today. Endometrial cancer today simply follows ovarian cancer. That may not be true in the future as we learn more and more about the molecular drivers of these cancers. And unfortunately, with cervix cancer, we haven't moved beyond cisplatin. Perhaps, again, there may be something here with immunotherapy, but that remains to be determined. So my comments will be focused on ovarian cancer because of the data, but that doesn't mean we don't have critical needs in other gynecologic cancers. I should also acknowledge that I will be speaking a little later on one critically important component of precision cancer medicine. Of course, that is the PARP inhibitors. So I will briefly refer to them in this talk. This is more of an overview, and then focus more on the PARP inhibitors a little later this morning. So I think it's very important when we begin to talk about precision cancer medicine, which I need to explain now what I mean by that, because there is and has been tremendous misinformation about this by many academics, predominantly in the United States, who have described precision medicine as an event. In other words, if there is a randomized trial that looks at a molecular targeted approach versus a standard of care approach, and it doesn't show an improvement, that shows precision medicine doesn't work. Wrong. Whether these individuals simply don't understand, which is possible, or they simply have a bias, which is also possible, is not clear. But it's important to point out that precision cancer medicine is not an event. It's not an outcome. It's a process. Precision cancer medicine simply refers to the idea that we are attempting as we move forward, and this is the future. Not a question about it, no debate here, if you understand it. The goal is to be more precise. Instead of using, what I'll talk about in a moment, are cisplatinums of the world, which are very effective and remain very effective in ovarian cancer, which are non-specific, simply hit targets, multiple targets. We're trying to be more precise with our treatments, both to improve efficacy and to reduce toxicity. That's all that is meant by precision cancer medicine. Historically, in ovarian cancer, we had active drugs. And in fact, outside of the hematologic malignancies, particularly lymphomas, ovarian cancer was the first malignancy, solid tumor certainly, again adding in germ cells to some extent, rare cancers. Ovarian cancer was the first tumor that was shown to be impacted by cytotoxic therapies. Now, of course, now I'm going back into the late 1940s, early 1950s, even the 1960s. And the activity of these drugs, alkylating agents, melphalan, alkeran, cyclophosphamide, were very real, but they were modest. And there was often significant toxicity associated with these agents because we often didn't understand how to use them. And then when cisplatin was introduced onto the scene, we had much more effective therapy, but also far more toxic treatment. And as a result, it was perfectly appropriate. In fact, it was the only response to the question of, is there value, is to prove an impact on survival. So randomized phase 3 trials were developed and conducted in many tumor types, including ovarian cancer, with the primary goal to show you impact survival. So I'm going to comment on the issue of trials as I move forward briefly, because it does impact on our ability to be able to demonstrate the benefit of a targeted precision medicine approach. It was also true at that time, and quite frankly up to the relatively current time, that second-line or third-line therapies, whatever you want to call them, had marginal effectiveness at best. Therefore, if you conducted a trial, let's say of cisplatin plus cyclophosphamide versus cisplatin and paclitaxel, of course the landmark study conducted in the late 1990s, which changed the standard of care in the management of ovarian cancer, it was appropriate at that point to look at overall survival as an endpoint, because what you did after that basically, unfortunately, had essentially no impact. And finally, and that really gets to the point of this lecture, epithelial ovarian cancer was traditionally, not today, viewed as really a single entity, certainly from the perspective of systemic anti-neoplastic therapy. So you can conduct a trial. You can take everybody with this entity called epithelial ovarian cancer, put them on the same trial, do randomization, and see if you impacted outcome. Again, traditionally, that was overall survival. The slide is, okay. The world has changed, very fortunately for our patients, and rather dramatically. And the speed of that change is only accelerating. Today, it is unquestionably true, not a debate about it, and if someone doesn't agree with that, we can talk about it later, that there are multiple therapies that now impact outcome in ovarian cancer. Multiple lines of therapy impact. Patients are living longer. I didn't say more are being cured, they're living longer. And appropriately, ovarian cancer is increasingly viewed as a very serious but more chronic disease process. Now, unfortunately, I cannot claim that's true for all patients, but an increasing percentage of patients, this is the nature of the illness. And we've also very clearly learned that certain subtypes are basically minimally, if impacted at all, by our standard therapies, including clear cell and mucinous carcinoma, where we really have no treatments of effectiveness. Again, demonstrating the lack of homogeneity of this illness. And finally, we are increasingly learning about the relevance of molecularly based, precision medicine strategy-based, more targeted therapies. I'm going to talk later about, of course, the BRCA mutation identifies a patient population where PARP inhibitors are relevant. Again, I'll come back to that. This concept of homologous recombination deficiency signature identifies a population, or may identify a population, that is to be determined, where PARP inhibitors are relevant, and again, a very precision medicine-based targeted approach, where it has now been unequivocally shown that in a patient population that has microsatellite stability insufficiency high, that is a laboratory-based measurement, identifies a patient population where, regardless of the tumor type, and in the United States, the FDA has now approved checkpoint inhibitor, one, and soon it'll be at least a second one, that is allowed to be given with response rates now of well over 40%, with many of those responses lasting for a year or longer, solely on the basis of this molecular target. And in ovarian cancer, that is 2% of the population, data published recently in Science, for the treatment of checkpoint inhibitor. So I think you can get my drift. 2% of ovarian cancer patients. How would you ever conduct a randomized trial of any kind looking at survival as an outcome when you're dealing with a precision medicine-based targeted treatment strategy that affects 1 out of 50 such patients. And yet, that's the kind of response rate, that's the kind of duration of response you can achieve. So now to go back to actual data in ovarian cancer to show the importance of the point that I'm trying to make and the problem we have when we are looking at this issue in gynecologic cancers, which are, fortunately, uncommon malignancies, even ovarian cancer. So here is a classic paper that I hope will bring to your attention the issue that I'm trying to deal with on behalf of our patients. Trastuzumab, Herceptin. I know in the breast cancer sections here where there's probably lots of conversations about HER2 and even trastuzumab, although obviously there are other agents now that have come on the scene with this particular molecular target. In fact, some of you may know, in the original paper in Science from Dr. Dennis Slayman that looked at the question of the relevance of HER2 in malignant disease, that original paper included both ovarian cancer and breast cancer. Dr. Slayman noted that patients with HER2 overexpression in both tumors types had poor outcomes overall compared to those individuals that did not overexpress HER2. The trajectories, however, for the development of anti-HER2 therapies in those two malignancies could not have been more different. And the reason is that this is a relatively common abnormality in breast cancer, and it turns out, and I'm going to show you this in painful detail in a moment, most uncommon in ovarian cancer, but still almost certainly relevant. So the only study that's been conducted in ovarian cancer that is quoted or noted is the study I'm going to show here. This was reported more than a decade ago in the Journal of Clinical Oncology. The Gynecologic Oncology Group in the United States decided to do a phase 2 single-agent trial. And as you can see, they looked at over 800 individual patients' tumors to find 95 that had even 2+ expression. Now, in those, obviously those days we were talking about a different molecular platform for measurement, but nevertheless, we can certainly translate 2+ into probably doesn't have a lot of relevance, or 3+, there may be relevance, but because there was such a small percentage of patients, they lumped them all together in this trial, and 54 patients were treated in this phase 2 study. 41 patients successful for response, and the point I'm trying to make is shown right here, where you see there were only 14 tumors, 14 patients on this trial. That is less than 2% of the 837 screened. Only 14 of those tumors overexpressed to a significant extent the HER2 receptor. In fact, there was 1 complete response and 2 partial responses in this trial. Of course, you would label this study as being negative, right? Wrong. This drug died in ovarian cancer because of the inadequacy of the trial design. I cannot tell you if the 3 responders were in those 14 patients because it was never reported. Nobody thought it was important to look at this. My point is, how many drugs are dying in ovarian cancer and endometrial surface cancer because of this inadequate design, inadequate approach, inadequate way of looking at molecular targeted therapy and precision medicine? Here is even a more painful story, because I can give you the bottom line. Gefitinib was looked at in a phase 2 trial, again conducted by the Gynecologic Oncology Group, published in Clinical Cancer Research in 2005. 27 patients were entered in the trial, standard phase 2 format. As you can see, there was standard toxicity, a 4% response rate. Clearly a negative trial, right? In fact, approximately 10% of the patients had overexpression of EGFR, which is what they were looking at, because there were those at that time, remember back even in lung cancer, the original hypothesis was that simply overexpression of EGFR was the relevant biological target, just like it was for HER2. And then, of course, in landmark work, it became very clear, and of course, we now know standard of care in lung cancer, it has nothing to do with overexpression or simple overexpression, it's particular mutations. So in this trial, there was one responding patient, one and only one, a negative trial, right? Except for the fact, the incredibly disturbing fact, and I hope you are as disturbed as I have been, they actually analyze the mutation status of all of the patients on this trial. In fact, they looked at 57 patients, sort of including patients that were not able to go on the trial, and 2 patients had a activating mutation of EGFR, so that's 3.5% of ovarian cancer patients, if that is seen as sort of the standard that you might see. Clearly inadequate to ever do a trial, clearly inadequate for you to ever look at that routinely in ovarian cancer, but the one responding patient in this trial, in fact, had an activating mutation in EGFR. Is this a negative trial? Absolutely not. This is precision cancer medicine in action, but I would dare say there won't be one of you in this audience would think about EGFR mutation when you have a patient with ovarian cancer you're considering for therapy, when you're talking about 3.5% of patients. And of course, if you knew the data, you would read this as a negative trial. So what we need to do, as I conclude, we need in ovarian cancer and the gynecologic malignancies to figure out a completely new paradigm for evaluating efficacy. There are a number of approaches that have been proposed. Simply high objective response rate, as was used in the MSI high story. Basket type trials that would include ovarian cancer patients, endometrial cancer patients, cervix cancer patients. Perhaps the prolonged time disease progression, objectively measured, compared to valid historical controls, or, in my opinion, what the future will be, is having patients serve as their own controls and looking at longer time to disease progression for that individual patient based on their prior treatment history, looking at the natural history of that individual patient. This concept was described in a truly landmark study by Von Hoff et al. In the Journal of Clinical Oncology in 2010, study known as the Bisgrove trial, where they demonstrated the benefits of that approach and that type of analysis. So that's another strategy. Of course, in each of these situations, it will be important to acknowledge the toxicity of the therapy, its impact on quality of life, and this must be acceptable. So I'm not talking simply about the question of therapeutic efficacy as measured by these parameters. You've got to also have that associated with toxicity and quality of life. And finally, I end with the critical need for biomarkers, which of course is what drives the concept of precision cancer medicine. We can no longer accept the concept that drug development is a lottery. And I'm very serious about this. That's what it is. Pharmaceutical company does a large phase 3 randomized trial, P is less than 0.05, they've won the lottery. They can get the drug approved and use it, charge what they want, that's the system, it's not anyone's fault, and simply give it to a large number of patients who are not going to benefit. We cannot do this any longer. We need to figure out who benefits, how much they benefit, and for how long. We need to come up with novel study designs looking at real-world treatment data, including the question of cost-effectiveness for our treatment paradigms, including measures of efficacy, toxicity, patient-reported outcomes, impact on quality of life. And I use as a negative conclusion the fact that in ovarian cancer, data from multiple checkpoint inhibitors to date demonstrate extremely unimpressive, and I'm being kind there, data relative to efficacy in phase 2 studies. In fact, that makes these drugs equivalent to nothing but better than topotecan in ovarian cancer. But at a cost, of course, that's about 1,000 times greater. And yet, phase 3 randomized trials have already been started by multiple companies across the board looking at checkpoint inhibitors without any concept of let's try to figure out what the biomarker is for the potential benefit in that 10 to 15% of patients. We don't know the drugs, we don't know the doses, we don't know the schedules, but we're always doing the phase 3 trial, already doing the phase 3 trials. This paradigm has to change, and I thank you for your attention.